It's my pleasure to introduce Arun. He's a PhD scholar in the Infectious Diseases Program of the Mississippi State University's College of Veterinary Medicine. Arun earned his Bachelor's of Fisheries Science from Kerala University of Fisheries and Ocean Studies in Kerala, India, and a Master's of Fisheries Science with a focus on aquaculture from the Central Institute of Fisheries Education in Mumbai. His dissertation work focuses on the epidemiology and phylogeny of ictolurid herpes virus 1 in U.S. farm-raised catfish. So, Arun, you can take it away.
Thank you, Dr. Phelps. And I just realized this is my third AFS presentation, and what says that we didn't had much opportunity. It's been a while. I guess everybody met due to the COVID, and I'm really thankful for the organizers because probably this was the only opportunity for us masters and PhD students across US to present our work. So today I would be talking about diversity of channel catfish virus from US farm-raised catfish and virulence assessment in channel and hybrids. Okay, so as you all know, the catfish industry is the largest aquaculture industry of USA, which is mainly located in Mississippi, Arkansas, Alabama, with a 398 million annual sales. A lot has changed in the industry. Currently, the hybrids are taking over the production system with a 60 to 70% of processed catfish in 2020. So next, today our topic is channel catfish virus. As you all know, channel catfish virus is the only reported viral disease in the catfish industry of USA, which mainly affects the fingerlings, and mortality can reach up to 100%. Vertical and horizontal transmission is possible, and the stress and the temperature can influence the possible outbreaks. And of course, the clinical signs, including distended abdomen, hemorrhagic area, and slight exophthalmia. Now, this is where I started my PhD project. So in our lab, in a pilot study, there are several CCV isolates has been analyzed using RFLP, and 3 distinct groups has been identified. So my work was to characterize them as genome sequencing, as well as study the virulence in channel catfish as well as the blue catfish. So my first objective was to assemble the sequence and assemble 20 CCV isolates, which we have collected from 1987 to 2011. So here the problem is, in general, short reads, which is Illumina, which can be 150 base pair length. If we use those reads or the sequencing technology, usually the herpes virus genome assembly would be often incomplete or broken. So this is a technical issue which we were facing. However, the new technology, nanopore technology, is available right now, which is long reads. I have a reads which can be 50 KB reads. So the total CCV genome is 134 KB. So I got the reads which can be almost half of the entire genome. So this technology, in combining with the short reads, can help us reference quality genome. So the long reads with a high error and the Illumina short reads which are pretty accurate, this combined use can help us to solve these repeats. So this is a small workflow which I have developed. So nanopore reads, we do some small pre-processing as well as the Illumina. After the pre-processing, we use an assembler, hybrid assembly, the assembler is Mazurka, which can take both the long reads and short reads at the same time and produce an assembly. Later on, I use a Samba, another package for the scaffolding. There are times I have to do some manual curation also. Later on, any other errors in the assembly is fixed with the polishing, with a long read polisher Medaka and a short read polisher Poly Polish. So now I got 20 reference quality genomes of CCV. The first work I did was to perform a variant analysis, which I separated into two categories. Structural variants, which are greater than 50 base pair length, and the SNPs and indels, which are less than 50 base pair. And here is a protocol I have followed. So it's basically different packages. I use it and I write some Python or shell program to run those. Coming to the results, the first one is structural variants greater than 50 base pair. Here, what I did, I called structural variants in each CCV isolate. Then I derived a population level genotypes. So these are the population level genotypes, and these are my samples. So for example, this is a genotype which is only present in blue catfish alloherpes virus. So now I got a map of structural variants, the population level, at a population level, based on 20 isolates, which is collected from 1987 to 2011. So an interesting feature, for example, blue catfish virus, which we described and we proposed it might be a new species based on our immunogenicity and virulence assessment. For example, here, as you can see, there is a 60 base pair insertion happening genotype, which is only present in blue catfish virus and absent in all other CCV isolates. Similarly, as you can see, this is a bit complex and a lot of data is there. This is for SNPs and indels for blue catfish virus as an example. The peculiar feature is here, as you can see, in blue catfish virus combined to the type strain, which is ATCC type strain. You can see there are variations or SNPs or indels, but there are really conserved regions too. So that is one conclusion I can say right now based on the SNP and indel analysis. So in the next step, so we developed a diagnostic method for two variants. So based on the initial RFLP analysis pilot study, we identified three genotypes: RFLP1A, RFLP1B, and the blue catfish virus. So in this objective, what I did, I developed a PCR assay to differentiate RFLP 1A and 1B. For that, first we designed 6 primers and probes for each RFLP group. Then I've used a cyber green assay to see whether there is any non-specific reaction. Later on, one set of primers which produces robust amplification was selected, and which was further tested with EMM, which is a TACMAN environmental master mix in a duplex format with a spread as internal positive. So the regions I have used for ICHV1A is aura 50, and for ICHV1B it's aura 49. So then I validated our assays according to the MIQE guidelines. So our efficiency was always between 90 to 110%. And the linear dynamic range of the assay was tested over 7 orders of magnitude. And in order to, we had one more objective, which I'll be coming next, which is to detect the latent forms of ICHV-1A and 1B. So in order to ensure maximum detection of latent virus, we wanted to use a high amount of the DNA extracted from the fry. So, but we want to make sure that, say, 1 microgram of DNA in the PCR mix would not inhibit the reaction. So, in order to test that, we have used different host genomes like CCO, cells, tilapia, koi, DNA, and we performed a spike assay, and we found that up to 1000 nanogram, there is no inhibition is evident in our assay. So we have decided to use 1000 nanogram or 1 microgram DNA in our PCR. So then one more issue which we faced is for the latent virus detection, as the cycles, some of them shows up after 35 cycles. And so in some cases, for example, some of them which shows up at 39 or 38, when I run the triplicate, all the 3 wells may not be positive. So which might be due to the extremely low copy number present in a latent DNA. So we developed this criteria saying 2 or 3 wells out of 3 is positive, 2 or 3, we will call it as positive. If only 1 well is positive, we'll call it as negative. And we used a Biorad CFX manager program to set it as, call it as automatically. So for the latent fry detection, what I did is I collected fry from different hatcheries. I did 3 sampling from each. I took 60 individual fry and extracted the DNA and performed the PCR. So the prevalence of ICHV-1 genotypes in the catfish industry, I had 6 hatcheries, and there is 42% samples were positive across the 6 hatcheries. In that, RFLP-1A genotype was slightly more prevalent than RFLP-A. So when we look at the hatchery level, the prevalence can range from 0 to 96%. In this case, it's 96% positive, and this is 90%. So what I meant to say, it really varies between the sampling and within the hatchery, and there is some variation among the channels and hybrids too. My next objective was to assess the virulence of ICHV-1A and ICHV-1B. Sorry, but the figure might look a bit complex, but it's simple. I took some channels and hybrids exposed to 3 isolates of ICHV-1A and 3 isolates of ICHV-1B. And I pooled the survivors, kept them at 26 degrees Celsius for 28 days. And ICHV-1A survivors were re-exposed with the A-type strain and a B-type strain. And it was compared with the 9. So in this, as you can see, the survival curves of channels and hybrid infected with the ICHV-1A. So there is a significant difference between ICHV-1A and B, and ICHV-1B seems to be slightly more virulent compared to ICHV-1A. In this case, it's the same thing. We are comparing between the channels and hybrids. There is only slight difference in the survival of channels and hybrids. So I would say there isn't any even though there is a slight statistically difference is there, but I would say biologically there isn't much difference. Similarly, this is a survival this is a reinfection study. In this, our ICHV-1A survivor re-exposed with A strain and B strain. Similarly, B survivor channels. So in this case, as you can see, these are there is a significant difference between the survivors and the naive channel catfish. So this says there is a cross protection effect is there in channels which are previously exposed to either ICHV-1A or ICHV-1B. Similar result in hybrids. Similarly, the hybrid survivors which are previously exposed to ICHV-1A or 1B had a significantly higher survival compared to the naive hybrid catfish. So as a summary, our project identified two discrete ICHV-1 lineages. Significant protective immune response in catfish that survive initial exposure of ICHV-1A or ICHV-1B. Rapid molecular confirmatory test to identify ICHV-1A and ICHV-1B variants is developed and validated. The prevalence of ICHV-1 ranged from 0 to 96% in the channel and hybrid catfish fry. And we got a reference quality 20 genomes of ICHV-1. So this genome information, the prevalence data and the virulence assessments can laying a foundation for future work investigating ICHV-1 variants role in CCVD in catfish aquaculture. So I would like to acknowledge my committee members, my major professor, Dr. Hansen, co-major professor, Dr. Griffin, my committee members, Dr. Weiss, Dr. Perkins, and Dr. Walsek. Then our team, Dr. Walbieser, and all other our technical staff and the scientists who has helped me in this project. So I would like to thank our sponsors, ICAR in New Delhi, India, USDA, and CVM and WFA department for their generous support to complete my project. Thank you.
Thank you, Arun. That's a great presentation. Folks, if you've got a question, feel free to drop it into the chat box or unmute yourself and ask Arun directly. We've got a couple in the chat here that I can read to you. One from Eva first, that you discussed the detection of the latent virus. I may have missed a crucial comment regarding your analysis, but are you assuming the virus is latent in these cases? Correct me if I'm wrong, but if you're extracting DNA and running qPCR, how would you ensure the virus is latent versus non-symptomatic or early shedding?
Okay. There is two parts in the answer. First one, we are directly collecting the fry from the trough, which after the hatching. So they are not exposed to the pond environmental condition, which rules out any horizontal transfer from the ponds. Now, the second case, there is we can differentiate between the latent and active based on the CQ values. So I would say anything which is above 32 CQ would be latent, and below 32 would be an active infection. And this has been already, Thomson 2005, they kind of worked on that, and we in our case also I had the same, and so I would say below 32 could be an active infection and above 32 would be a latent infection, give and take.
Okay, thank you. Eva, you can follow up if you've got another question on that. You've got another one here, Arun. First, a compliment on a great presentation. Can you give us more information on the channel catfish strain, the USDA 103, and the type of hybrid catfish used in the study?
This is not exclusively a USDA strain. What we have used is an industry pool. So why we used that, there are earlier papers between the strains of channel catfish or hybrid catfish, there could be a variation in the virulence. So in order to remove that effect, what we have used is an industry pool. So we got the fish from different hatcheries, which has used different populations of brooders, and we just combined them.
All right, I see your hand up, Mikolash.
Hi, Arun. Great presentation. You had a difference in between the two, let's say, strains of the virus in virulence, and you did also some genome sequencing.
Mm-hmm.
Can you link any broken genes or genes which are longer in your genomes to the increased virulence, or you were not looking at this yet?
Yes, as you said, we are looking into that. So you have seen I got up to the variant structure. So we know what is varying, what are the regions which are varying between the isolates. Now, my next step would be to figure out which would be the causing this variation in the virulence. So yeah, that is a work in progress.
Any other questions out there? All right, if not, virtual round of applause to you, Arun. Great job.
Thank you.